I'd like to start by firstly thanking the organizers for this fantastic meeting, and it's been a pleasure listening to all the talks. We've heard a lot about archaic introgression, and what I'm going to talk today is about the potential impact of archaic introgression, specifically Neanderthal introgression, on human biology. And so the reason why we think it's plausible that Neanderthal introgression has had a substantial impact is because of the fact that there was this big split between Neanderthals and modern humans of the order of hundreds of thousands of years, during which there were mutations that accumulated on the Neanderthal lineage, which entered into the modern human gene pool around 50,000 years ago, and then were acted upon by evolution. So one way to see this is to look at a different perspective, which is going beyond this 2% fraction of genetic ancestry that's inherited from Neanderthals. If instead we ask a slightly different question in simulations as to what fraction of segregating sites at the time of introgression trace their ancestry to Neanderthal introgression, we get a substantially larger proportion, about 10% of segregating SNPs at the time of introgression. So that's a substantially larger contribution. So to try to understand the impact of Neanderthal introgressed alleles, one class of approaches has been to look at the impact of these alleles on fitness. And so there's been several studies which have shown that there is this reduction in Neanderthal ancestry at regions of the genome that are under strong selection. So on average, the picture is that Neanderthal alleles are deleterious. However, there are other regions of the genome where Neanderthal alleles are adaptively beneficial, as seen by the fact that they are at substantially higher frequencies today than about 50,000 years ago. So beyond the impact of introgressed Neanderthal alleles on fitness, there's a question of whether there are specific phenotypes that are impacted. This has also been addressed by several groups by looking at genome-wide association studies. So this was an early paper from a few years back, which looked at type 2 diabetes and identified an introgressed risk factor that was associated with increased risk for type 2 diabetes. And more recently, there's been a work that has shown that these introgressed variants have also been contributing to increased risk for severe COVID-19 response. So despite these examples, it's generally been challenging to study systematically the impact of Neanderthal introgression on complex traits. And the reason for this is twofold. Firstly, as we have heard from the first talk in today's session, complex traits are influenced by large numbers, often thousands, tens of thousands of genetic effects, each of which are quite small. The second is introgressed Neanderthal alleles are rare on average. So if you look at, say, the 1,000 genomes European, it's a couple of % in terms of the derived allele frequency. So the upshot of all of this is the power that you need to be able to see these kinds of effects is fairly small. So we were thinking about how we could go about studying introgressed alleles with sufficient power. This was many years back. And at that point, we became aware of this study, the UK Biobank, which was collecting genetic data along with thousands of phenotypes across 500,000 UK individuals. So this seemed like a perfect setting in which we could try to study the effect of Neanderthal introgression. So we got in touch with the group that was designing the UK Biobank array at that point. And it turned out that they had some space to add interesting content onto this array. And so we asked them whether we could add SNPs which tag Neanderthal ancestry. So essentially, we were picking out these SNPs, mutations that lie on introgressed Neanderthal haplotypes. We call these Neanderthal informative mutations, or NIMs. So it turned out that we started out with about 100,000 NIMs identified in 1,000 genomes European populations from these Neanderthal ancestry maps. Many of those were already tagged by the existing content of this UK Biobank array. And then we added a few more so that all the NIMs that we had identified were tagged by the array at a sufficiently large R-squared threshold. Once the individuals in the UK Biobank were genotyped, then there was an imputation that was done that substantially increased the number of SNPs that were covered. And once we also looked at the imputed SNPs, we found that we could recover all of the original set of about 100,000 NIMs. We also added additional NIMs which were in strong LD to this to get about 200,000 NIMs with which to analyze the UK Biobank data. So essentially, the data that we were looking at has about 300,000 individuals. These are unrelated white British individuals in the UK Biobank with imputed genotypes. As I said, we care about alleles that are rare. That's the bulk of these introgressed alleles. So we were looking at SNPs with minor allele frequencies of greater than 0.1%. So that's about 8 million SNPs, of which about 200,000 are these NIMs. And we looked at about 96 phenotypes, spanning 14 broad phenotypic categories. So the kinds of questions that we were interested in, and this is not something that we have done alone, there have been other groups that have also looked at similar types of questions, are along these lines. So the first question is we'd like to understand what is the contribution of these NIMs as a whole to phenotypic variation. So this is related to the notion of heritability. Then the question is whether the contribution of these NIMs to phenotypic variation is more or less than non-introgressed modern human SNPs with a similar allelic age. So if you have two mutations, one is Neanderthal, one is modern human, born at the same time, and then they are segregating in a modern human population, do they contribute similarly to a phenotype? And finally, can we pinpoint regions of the genome and NIMs that actually modulate complex traits? So the first question, as I said, relates to this notion of heritability. So we call this NIM heritability. The second one is related to the notion of heritability differences between NIMs and modern humans. So we call this delta H squared. And finally, the final question is related to this notion of fine mapping. So we call that NIM fine mapping. So we first looked at this notion of estimating heritability. And to do this, we use a very effective tool from complex traits genetics, which is this notion of variance components models. So the key idea of variance component model is because effect sizes are really small, it's challenging to first identify these individual SNPs with effects and then ask what's their contribution. So we'll set up a model which averages over the identities of these variants and directly tries to estimate whether they have an effect. So the model looks something like this. We write down the phenotype as a linear combination of our genotype plus some noise. And because the number of SNPs we are analyzing is still substantially larger than the samples, we're going to put some constraints on the effect sizes of these SNPs. So typically, we would say that the effect size of a SNP comes from some distribution with mean 0 and some variance. And this variance parameter is the parameter that we care about. So that's related to heritability. Of course, that's the vanilla model, and we can make it substantially more general. For example, in our setting, we might have one variance parameter if a SNP falls within a NIM, and another variance parameter if it falls outside. And now we are interested in estimating and comparing these different variance parameters. So this type of an approach has been incredibly influential in human genetics. So this is a paper which applied this type of an approach to different types of functional regions in the genome. And the key quantity of interest here is there an enrichment of heritability, a genetic signal, in specific functional regions of the genome. So the statistical model is clear, but the problem is one of scale. So the key issue is we have to apply this to this large data set, 300,000 individuals, 8 million SNPs. And so these variance components models are extremely challenging to apply at this scale. So another interest in my group is how do we build statistical models to allow us to infer genetic architectures of complex traits, which can actually leverage these large data sets. And the key idea is we're going to work not with this gigantic genotype matrix, but with a sketch of this matrix. So we're going to take this genotype matrix, and we multiply it with some number of random vectors. So that's a parameter of this estimator. And so the smaller the number of random vectors, smaller the number b, the smaller your sketch. And so that's going to be more efficient to work with. So that's easy to see. But what is not obvious, and it's a bit of a surprise, is the accuracy of this method is actually quite good for pretty small values of this sketch. So we can actually get accurate estimates of quantities like heritability while being extremely computationally efficient. So this is a method that's been published a couple of years back, and I won't spend too much time on the experiments, but here we compared this method, which we call randomized Haseman-Elston regression. So this goes back to the fact that this is a modification of a classic method that comes out of quantitative genetics, except it's a randomized version. And what we find when we compare it to methods that have been developed, which are scalable, so these are methods that are what we call summary statistic-based methods, these randomized Huysman-Elston type approaches are quite accurate, while being extremely efficient, so they can be applied at the scale of UK Biobank. So now we have a methodology that we can apply, potentially to answer and estimate NIM heritability. But before doing this, we set up some simulations. So we started out with whole genome simulations. So we take our matrix, we simulate these complex traits, polygenic complex traits, and we ask, can we estimate heritability accurately in this setting? So we did a lot of different variations. We considered traits with different heritabilities, different polygenicities, so different numbers of causal SNPs. And in the complex trait human genetic literature, it's becoming clear that these kinds of traits often have patterns of variation that depend on whether a SNP is common or rare, and on the LD patterns surrounding a SNP. So we investigated lots of different possible combinations. And what we see here is we are applying this method to estimate NIM heritability, and we are asking how close is our estimate to the truth. And so if it falls within this band that we have here, that's hard to see, then this method is calibrated. So what turns out to be the case, and maybe it's not obvious, is the method is not quite calibrated the way we've applied it. We can now ask the same question about testing differences between heritability at NIMs while comparing it to heritability at modern human SNPs. So the question we're asking is, if I look at one random NIM and estimate its heritability, how does that compare to the heritability estimated at one random modern human SNP? So the key thing to note here is in these kinds of simulations, we have set it up so that the simulations are completely blind to the identity of whether a SNP is a NIM or a modern human SNP. So which means there should be no difference in these simulations. And what is evident here, and quite disturbing, is that there is quite a bit of bias in our estimates of these differences in heritabilities. So the reason for this, it turns out, is NIMs have quite different properties compared to modern human SNPs. So here we are looking at two of these. So here we are, on the left, we have the minor allele frequency of NIMs compared to modern human SNPs. And what we see is, in general, NIMs have lower minor allele frequencies compared to modern human SNPs. So this is consistent with the fact that these were introgressed around 50,000 years ago with an ancestry fraction of around 2%. In addition, we can look at the LD patterns around the SNP. So we can ask, on average, what is the sum of the R² metric of this SNP to SNPs nearby? And it turns out that NIMs tend to have higher LD as measured by this quantity called LD score compared to a modern human SNP. So on both these metrics, there are differences in these properties, which we hypothesize is the reason why we are seeing these biases. So one way to solve this in this framework, given its generality, is we not only fit our variance components model to whether a SNP is a NIM or a modern human, but to also these additional annotations. So we add in these MAF and LD annotations in addition to the ancestry annotations. And so we tried a few different combinations. So we tried looking at ancestry plus MAF, ancestry plus LD, and finally ancestry plus MAF and LD. And it turns out that adding both the ancestry and the MAF and the LD annotations leads to accurate estimates of the NIM heritability. And importantly, it also leads to accurate estimates of the heritability differences. So when we apply this kind of a technique on real data, we add both ancestry in addition to these MAF and LD annotations. So we then applied this approach to our 96 UK Biobank phenotypes. So this is an estimate of the NIM heritability across these phenotypes, grouped according to their categories. And the key takeaway is these phenotypes have a NIM heritability, which is positive, statistically significant, but fairly low. So averaged across these phenotypes, we estimate a NIM heritability of the order of 0.1%. It's statistically significant. And this is consistent with the polygenicity of the phenotype. And so if you ask if there's 2% of the genome that is coming from a NIM and you have a heritability of 10%, then this is roughly what you would expect. Then we looked at the differences in heritability between NIMS and modern human SNPs. And here, what we find is when we look at this delta H squared, consistently across phenotypes, there is a negative delta H squared, which is telling us that heritability at these NIMS is negative compared to modern human SNPs. So NIMS explain less heritability compared to a modern human SNP after matching for allelic age as given by the MAF and the LD properties. Importantly, we don't find any evidence for elevation in NIM heritability across any of these traits. And so this observation of a depletion in NIM heritability is consistent with purifying selection on NIMS. So this is, again, consistent with a number of studies that have shown that on average, NIMS are under purifying selection after having entered the modern human gene pool. Okay, so now I'll move to looking at pinpointing the regions that are associated with phenotypes. So the way we can do this is by doing a genome-wide association study. So we looked at each of our NIMS, and we performed a standard genome-wide association study with our set of 96 phenotypes. We looked at a very stringent p-value threshold for significance. So this accounts for both the number of phenotypes we are testing and the number of SNPs that we are testing. And what we find is about 300 unique NIMS that are associated with each of our phenotypes. The problem with using this to conclude that these NIMS are actually associated with the phenotype is that this could also arise due to the fact that some of these NIMS are in LD with modern human SNPs. So this was a point that was made quite eloquently in this paper from Decode Genetics. And the key idea is if you have a modern human SNP, like these red arrows, which are truly causal, it is possible that a NIM that is in LD with a modern human SNP appears to be more significant than either of these modern human SNPs. And so that's not the truly causal variant that we're interested in. So to deal with this problem, again, we turn to the work that has been done in the statistical genetics literature. And so there's been intense work at trying to deal with this issue of how do you determine whether a SNP is truly causal. So this goes by the name of fine mapping. And so what these methods do is they look at the association signal in a region, and they consider all possible configurations of causal SNPs. And they integrate over these possibilities to finally come up with what we call a credible set. And the interpretation of a credible set, a 95% credible set, is that with probability of 5%, you're guaranteed that a causal SNP lies outside this credible set. So again, when you're applying these fine mapping methods, at the scale of the UK Biobank, there are serious computational issues. And in this setting, we were fortunate to use a recent fine mapping method from Matthew Stevens' group called SUSY, which can actually be applied at scale. What we are interested in here is in asking the question of not whether just a single SNP is causal. We want to know whether in a given region are any of the NIMS causal for a phenotype. So we did this by setting up this pipeline where we start with NIMS that have been identified as significant in our GWAS. And then we LD prune these NIMS and construct regions around these NIMS, which include both modern human SNPs and the NIMS. We give these as input to SUSY, which gives us these credible sets. And to finally declare whether there is any NIM that is causal in this region, we require that a majority of the SNPs which lie within this credible set should be NIMS. So this gives us what we call credible NIM regions. So again, we evaluated this approach using our genome-wide simulations. And so we first compared the false discovery proportion of just using a GWAS approach to this fine mapping protocol. And what we find is this fine mapping reduces or halves the false discovery rate compared to just doing the GWAS. So the GWAS starts with a false discovery rate of around 30%, and the fine mapping reduces it to about 15%. So we applied this fine mapping approach to our set of GWAS associations. And what we find is we move from about 300 NIMS that are associated with phenotypes to about 100 credible NIM regions. So what we get is a region where we are confident that there is a causal NIM. What you'll see here is the number of phenotypes where we have these credible NIMs has fallen from about 64 to 47, and that's because of the trade-off that we are making in terms of power versus false discovery proportion. So there are some phenotypes where we find no evidence for a credible NIM. So these regions can be quite big. They can contain on the order of 50 NIMs, and the reason for this is the resolution of these credible NIM regions is determined by the length of the introgressed Neanderthal haplotype. So often, NIMs that fall on the same introgressed haplotype are in very tight LD, and so we don't have much resolution in being able to say which of those individual NIMs on a haplotype are actually causal. So this is a limitation of this approach, and so this is something that we are actually actively working on. So now that we have this list of credible NIMs, we can now try to understand what are the functional effects of some of these credible NIMs. And to do this, we annotated these credible NIMs using the software SNP effects. And we looked at lists of credible NIMs which were identified as having a high impact according to the SNP effects annotation. So here is an example of one such NIM. So this is a credible NIM, which leads to a loss of a start codon in this gene called COC10A. And COC10A is a gene that is known from prior studies to have mutations that lead to skeletal and muscular disorders. And so this credible NIM is associated with standing height. So that makes sense from the point of view of the functionality of this gene. The other interesting aspect about this credible NIM is it lies in this credible NIM region that you see here, where you have a set of linked NIMs, which are all associated with height. And this also overlaps another gene, STAT2, which is a notable gene because this was the first gene that was documented to have adaptive introgression from Neanderthals. And so STAT2 is a gene that has been also shown to be important for immune-related function. So here's another example of another credible NIM with high impact as defined by this annotation. So this is a NIM which is a non-synonymous mutation in this gene called AKR1C4. So this is a gene that is known to be important for metabolizing steroids in the liver. And what we find is that this NIM, based on our phenotypes, increases the levels of blood bilirubin. So this is an enzyme that is associated with liver function. Further, when we look at the GTEx data, we find that this is a NIM that is a splicing QTL for this gene, which lies upstream of AKR1C4. So we did this for several other NIMs, and so we have about 19 genes which are annotated to have these credible high-impact NIMs. And we find that 7 out of these 19 genes are known to have immune-related function. So this is consistent with prior work, which has shown that many of the genes where there is a substantial impact of Neanderthal introgression tend to have immune-related activity. The other genes that were annotated this way, they have functions related to DNA replication or damage repair, meiotic transitions, and neurological and developmental functions. So overall, the picture that is coming out of this type of an analysis is that there is a substantial contribution of these credible NIMs in genes that are related to immune function. And so this has also been proposed in other studies where this hypothesis is one where as modern humans leave Africa and move into these newer environments, having these introgressed alleles impacting these immune-related genes could have been an efficient way for modern humans to adapt to these environments. Of course, our results don't directly support this hypothesis, but it could be plausible. So just to summarize our results here, so this type of an analysis of combining biobank data with evolutionary genetic information allows us to understand the impact of introgressed Neanderthal mutations on complex traits. So the first thing we identify is a significant contribution to complex trait heritability. However, NIMs tend to be depleted in their overall contribution to complex traits relative to non-introgressed modern human SNPs, and this is consistent with purifying selection on introgressed alleles. However, what's missing, and something that we are actively working on, is trying to figure out whether this tells us something additionally about models of selection in the human population post-introgression. We are able to identify hundreds of regions in the genome where NIMs can credibly modulate phenotypes. So this uses fine mapping in combination with association studies. However, in all of these analyses, we are trading off power for low false discoveries. So likely what we are finding is just a small fraction of NIMs that are causal. Nevertheless, I think this is a good starting point for more detailed investigation in terms of their functional impacts. And so stepping back about how these kinds of data sets can be used to better study the functional impact of archaic introgression, the first thing to keep in mind is there are some challenges due to the distinct population genetic properties of these introgressed variants. So this is something to be aware of when doing these types of analyses. So the first challenge is the requirement of large sample sizes. The UK Biobank and similar such biobanks offer a potential approach to deal with that limitation. However, analyzing these biobank data sets brings with it additional analytical challenges. So a necessity here is biobank scale methods, which can actually leverage the power of these large data sets. With this, I'd like to acknowledge my group. So the people who did the bulk of this work, a very talented postdoc, April Way, who's now a faculty at Cornell, my PhD student, Chris Robles. I'd like to thank all my collaborators, the UK Biobank Array Design team, and funding sources. Thank you.